To start off with, so those of us in the acne and rosacea domain know that the skin microbiome has been a hot topic for the last couple of years. And imbalance, also known as dysbiosis of the skin microbiome, has been shown to contribute to inappropriate inflammation in several disease states. For example, in atopic dermatitis, Nakasugi et al. Recently showed that atopic derm patients have a relative deficiency of commensal staphylococcus species that can actually antagonize Staph aureus, causing Staph aureus to overgrow and the disease state to worsen. Barnard et al. Also reported that compared to healthy controls, strains of Cutibacterium acnes that were isolated from acne patients were more virulent. The role of the microbiome in rosacea, however, is still not very well understood. Demodex has been historically associated with the pathogenesis of rosacea, but recent studies have sort of been very inconclusive with regards, some not showing any correlation whatsoever. And it also seems like a lot of literature on the imbalances in various commensal organisms have been implicated in the pathogenesis of rosacea, but none of the evidence really seems so definitive. So we were particularly interested in the commensal staphylococcal species, particularly since Staph epidermidis was isolated from rosacea pustules in 2011. And there's a growing body of evidence supporting the ability of commensal organisms to influence health or disease states. Just as an example, Staph hominis was shown to produce a lantibiotic that is active against Staph aureus. Staph lugdunensis also makes a cyclic peptide that can kill Staph aureus. Staph epidermidis has been shown also to make a phenol-soluble gamma modulin that can inhibit both Staph aureus and group A strep. And Staph epidermidis was also shown to co-colonize with C. acnes and inhibit the inflammation that C. acnes cause by decreasing TLR2 production. We're also interested in the behavior of C. acnes as it's a well-established member of the facial skin microbiome that dominates sebaceous sites in the skin, as we all know. And some strains have been shown to exert a protective effect on the skin. In particular, C. acnes prevents colonization by other microbes because it breaks sebum down into free fatty acids and short-chain fatty acids, which lowers the pH and suppresses the growth of bad bacteria, such as MRSA and group A strep. And last but not least, Staph epidermidis and C. acnes have both been noted to be prevalent in skin samples from untreated rosacea patients. Now, topical ivermectin, commercially known as Soolantra, it's a safe and effective treatment for rosacea, and it's been reported to decrease Demodex mite burden and also decrease serine protease activity as well as cathelicidin production. And it's also been shown to be anti-inflammatory and to decrease cellular and humoral immune responses. But we wondered that if some part of its therapeutic effects might have to do or might be correlated with a shift in the populations of commensal skin organisms, specifically Staph epi and C. acnes. Like, how does it all tie together? And so in order to explore this question that we had further, we went ahead and carried out a single-site, 12-week, open-label clinical trial with 10 subjects aged 18 to 85 with papulopustular rosacea who had at least one papule and at least moderate erythema. They were treated daily with topical ivermectin for 3 months. We collected skin swabs from both lesional and non-lesional skin at baseline and after treatment, and then we extracted the microbial DNA and conducted qPCR to quantify the DNA abundance of each organism in the populations, and also used genus-specific 16S primers in order to measure the DNA abundance. And then we clinically looked at the number of papules and the assessment of erythema at each visit. So 16S phylogenetic analysis of the microbiome from a selection of 3 of the subjects before and after treatment in lesional skin is shown here. The relative proportion of the bacterial species is represented in the different colors. The yellow bars show Cutibacterium, which unsurprisingly dominates facial skin. The dark blue bars show Staph epidermidis, and the right graph in each pair here shows the species breakdown excluding C. acnes, just to get a greater differentiation of the non-C. acnes since it's so dominant. But overall, it does seem like treatment with topical ivermectin appears to increase the proportion of Staph epidermidis. And then these are similar graphs, as I previously explained, but with non-lesional samples. Again, there's a small increase in the proportion of Staph epidermidis in some subjects after treatment. And these graphs here show the absolute abundance of Staph epidermidis on the left and C. acnes on the right in lesional and non-lesional skin before and after treatment. We see a significant increase in the absolute abundance of both Staph epi and C. acnes in lesional skin, but not in non-lesional skin. Further, our results do corroborate that the absolute abundance of Demodex mites decreases in both non-lesional and lesional skin after ivermectin treatment. So we wondered how much of these quantitative trends that we were seeing in the DNA abundance was caused by, just live bacteria, dead bacteria, like what exactly was contributing to it. And so we went ahead and plated some of our skin swabs on mannitol salt agar in order to quantify live colony forming units. And this confirmed that treatment with topical ivermectin leads to an increase in live commensal staphylococcus in both lesional and non-lesional skin. We also observed that increase in Staph epidermidis was associated with clinical improvement. That is, the abundance of Staph epidermidis negatively correlated with both erythema and the number of papules on the patients. In contrast, we observed that a significant decrease in Demodex mites correlated with a decrease in the papules, but not with an improvement in the patient's redness. And then last but not least, we noted that ivermectin treatment increased the diversity of the microbiome community as measured by the Shannon Diversity Index, but not by the Chow Richness Estimator, which is more based on presence of rare species. And so in conclusion, we found that the ivermectin treatment increased abundance of Staph epidermidis and C. acnes in lesional rosacea skin, and also decreased abundance of Demodex mites. Increase in the Staph epidermidis was clinically correlated with a decrease in clinical erythema and papule count, whereas the decrease in Demodex mites correlated with improvement in papule count, but not with erythema. Diversity increased after treatment, and based on these findings, we thought that treatment with topical ivermectin may play a role in improving the symptoms of patients with papulopustular rosacea by modulating the skin microbiome beyond just decreasing Demodex.